hello everyone. My name is Kamila Muchowska. I am a research assistant professor, or in French, chargé de recherche, at the Institute of Supramolecular Science and Engineering in Strasbourg, where I work at the Laboratory of Chemical Catalysis directed by Professor Joseph Moran. So today I'm going to give you an introduction to the metabolism-centered approach to the origin of life on Earth. And I will also tell you a little bit about all the research that has been done at the Laboratory of Chemical Catalysis in Strasbourg that involves this approach to the origin of life. So before we get to science, we need to go back in time. And that is going back in time by about 4.5 billion years, when there was no life on Earth. So the planet Earth has just accreted, it has just formed, and it was very young, and obviously it wasn't uninhabited. But thanks to all the planetary scientists and to geologists and everybody who is involved in the research of the early forming planet Earth, we know what probably was present there on the young planet. So, for example, on the lifeless planet Earth, we know that water already existed about 3.8 billion years ago. We know that probably the atmosphere contained a lot of carbon dioxide, CO2. And we also know that the planet was very abundant in metals. For example, the core of Earth consisted and still consists of mostly molten iron and molten nickel. So these may make up over 80% of the core of the planet. So these are reduced metals, they're in the center of the planet. But the further you go outside, so throughout the mantle towards the crust, the more oxidized the metals become. So this creates a sort of redox or electrical imbalance throughout the entire planet on the planetary scale. So let's remember about that. It will be useful later. But all in all, so what we did have on the lifeless planet, the young Earth, was plenty of really small basic building blocks that can undergo geochemical processes. And obviously they did, and we will see what we can do with that later. So the origin of life on Earth, you could think that this is a philosophical question that mostly philosophers and some thinkers get preoccupied with. But it's not just a philosophical question, because if we want to say that we understand life, understand how cells work, how life works, we need to know how it came to be. So the general purpose of the research I'm going to tell you about today is how to have a deep understanding of the chemical foundations of living cells and how they became the way they are today. So my little disclaimer is that although very many astrochemists and astrobiologists theorize about life in outer space, how life can be created, how life could arise on other planets, other planetary bodies. What I'm going to focus on is life as we know it. So the only life that we know is on Earth, and this is what I'm going to focus on. So in other words, the specific question that we are trying to answer is how the geochemistry, which you have depicted on the left-hand side of the slide, underwent chemical processes such that allowed it to self-organize and become something that you have in this circular part of the slide. This complex pattern, this complex map that we call biochemistry, which is a depiction, a chemical depiction of what life does, all the chemical processes that life does today. So the area of science that addresses these questions and this problem that tries to explain it is called prebiotic chemistry. So this is this very messy and very complex picture is how we currently depict biochemistry. It's an incredibly complex map of chemical transformations, different molecules, and basically it describes everything that life does to keep life alive. So the two important things that you should know about this map is that it is incredibly well organized and it contains patterns. You can see some circular, sort of snail-shaped, vortex-shaped patterns, lines, squares. But you should also remember that this is not just a static pattern. This is not just like a mandala that you could fill with colors. This is something that keeps changing all the time. It remains in motion. It's a dynamic structure, a dynamic organization. So molecules become converted to one another all the time. Different processes occur all the time. So metabolism is complex, as you can see, but to us chemists, because I am a chemist, metabolism only uses a very small fraction of all the chemical mechanisms known to organic chemists. So in the central part of metabolism, we find about 6 or 7 reaction mechanisms. So this is very few. To me, as a chemist, this is quite surprising as well. So this means that life has a very particular way of making its molecules and of conducting its processes. So we wonder, like, why is it that? Why is it like this? Why has life chosen only these 6 or 7 different types of reactions to build all this incredible complexity? So before we move on, and this is something that I will do throughout this talk. We will, every now and then, we're going to stop and reflect on a definition, because we need to get the definition straight in order to understand this lecture. So metabolism in biology, according to a textbook definition, is a collection of different chemical processes that utilize building blocks, simple building blocks, which we can also call them a food set, to build up build up the molecules that take part in this living systems process. And these processes are also involved in the internal maintenance of the system. They use an external energy source to keep all the processes going. So our biochemical metabolism can be basically broken down into 2 types of reactions. So one type of reactions is the anabolic processes, which are the processes that build up molecules for which they need a source of energy. The opposite process is called catabolism. And this process uses the more complex molecules to break them down, and in this way we obtain energy. So these are the basic definitions that you should know. Now time for a more global outlook. So to get us in the right state of mind to look at the questions of the origin of life, we need to think about what life actually does on a more global scale. So when you look at the entire ecosystem, so what happens is that life in general converts geochemicals, simple geochemicals, such as carbon dioxide, hydrogen or nitrogen. It converts them into slightly more complex biomolecules. And these are, for example, fatty acids, amino acids, carboxylic acids. So these are biomolecules that are relatively simple. So what happens next? These biomolecules can polymerize, and they can polymerize into 2 different types of structures. So the third rung of this pyramid, third rung from the bottom, shows polymers that are polypeptides or, for example, proteins, and also enzymes. Enzymes are excellent catalysts. Another thing that can happen is the biomolecules such as ribonucleotides can polymerize to give us genetic polymers. And now if we think about where all these things came from, we encounter into something that can be called the chicken or egg problem. Because the way that the molecules on the second rung are made, so the simple biomolecules, they are made in reactions which are enzymatic, which means that they are catalyzed by enzymes. But the enzymes are higher up in the hierarchy. And how the enzymes are made? They are made from those metabolites. And how they are made, in what way, or the sequences, or the recipe for making them, this information is contained within genetics. So if we want to think about what came first, this presents sort of a paradox. So there are several ways that prebiotic chemists, or chemists who work on the origin of life, this can be approached in two ways. So the major two distinctions in the approaches to this. One hypothesis I want to tell you about is the RNA first hypothesis, so the genetics first. So these scientists present the view, they support the view that initially on the early Earth, before life, there were processes that led to the polymerization of genetic polymers, which then allowed for the sort of lower hierarchy to be built. So the genetic polymers allowed for the formation of peptides, of biocatalysts, eventually those simpler metabolites. The opposite sort of hypothesis is called metabolism first. So the metabolism first hypothesis follows the hierarchy from the bottom of the pyramid to the top. So it follows the order of complexity. So these scientists, amongst which you can also include myself and the laboratory I work in, so we support the view that geochemicals must have given rise to simple metabolites, which then started to polymerize to produce, for example, peptides, polypeptides, biocatalysts, and eventually they gave rise to ribonucleotides and RNA and genetic polymers. But why would we even think that metabolism originally self-organized from geochemicals on the early Earth rather than be a result of some pre-existing gene or genetic polymers programmed catalysis? Why should we support this metabolism first hypothesis as opposed to genetics first? Like, what is the deal? So this question touches on the very distinguishing feature of the self-organized systems. And this feature is that self-organized systems, complex systems, they emerge if we have some simple mechanisms that are superimposed. So several scientists, actually very many of them, investigated this problem from a mathematical or a systems biology point of view. And in short, this is a very difficult, area of science, at least to me. But in short, the message is that, for example, in those two papers that I show you here on the slide, please don't try to read them, you can look them up after the lecture, but the main message is that that the self-organization of metabolism is visible on very, very different levels of complexity. It is visible on a level of a simple organism and an individual cell, but it is also visible on an ecosystem level. So for the entire biosphere, it has been calculated, it has been modeled. So if you would like to delve into the details, I encourage you to look up these two papers and basically enjoy the systems approach to biochemistry and biology in general. But wait, did I just say that 6 or 7 types of reactions, chemical reactions, would give rise to such an incredibly complex structure as biochemistry? Is that even possible, that this huge map with thousands and hundreds or thousands of different chemical reactions and different transformations emerge just from 6 or 7 simple rules? It doesn't sound very realistic, does it? Well, it is not that unlikely that 6 rules give rise to an incredible level of complexity. So this example I'm showing here, it is obviously not very related to chemistry, nor is it related to biology. But take a game of chess. So we have 6 types of pieces, each of which can move in a specific way. So we have 6 types of movement that are allowed, and we have a space which is limited by the 64 squares of the chessboard. And even though the rules of movement are only 6, the possibilities are almost unimaginable. There is a number of 10 to the power of 123, which is called the Shannon number for chess, which mathematicians have calculated to be the number of different possibilities for this game. And the same analogy in the conceptual level, of course, with some which is a very big simplification, but I think it helps to understand how using only 6 or 7 different types of reactions we can obtain something as complex as biochemistry. Here you can see another illustration of this idea. There is an old school computer game called the Conway's Game of Life. And basically what happens in this game is you have a grid of black and white pixels, and you can apply some rules. You can program some rules. So most of the time, if you apply those rules, nothing spectacular happens. But sometimes a very specific set of rules causes this dynamic structure to emerge. And when you look at this from a distance, sometimes it starts to look like you're looking at a bunch of cells under a microscope. So, so this goes to show that, that in principle, for a complex system to emerge, you need to have several mechanisms that are superimposed in a specific way, and then you have this emergent behavior, a dynamic system that emerges from this. So in the Conway Game of Life, these rules are programming rules, right? And they're programmed by the programmer. But you could draw an analogy. If you take, for example, 6 or 7 different rules that are chemical mechanisms, and you superimpose them in a certain way, then a dynamic structure like protometabolism is also likely to emerge. So approaching the emergence of life is a matter of self-organization and of dynamic behavior. So this is what we have established. But life is also a dissipative process, and I'm going to explain what it means. So it's not a collection of static molecules, but it is a collection of processes which all together assemble into a big dynamic process that life is. So dissipative structures are open systems which are out of thermodynamic equilibrium with their environment. So in nature we have many, very, very many of such systems. And for example, we have a hurricane, we have a water vortex, we have oscillating chemical reactions like the Belousov-Zhabotinsky reaction, the chemical clocks. And all these systems are dissipative, which means that they are driven into existence when you have a constant supply of energy. So the characteristic dynamic structure that you see as a result of the dissipation of this energy appears because when the energy is accumulated, when there is a gradient that is accumulated, it becomes released through a pathway of least resistance. So this is a kinetic pathway of least resistance. And life is also like a dissipative structure that follows an organizational pattern similar to, for example, a hurricane. So the building blocks of a hurricane are air and water. But obviously, it's not enough to take air and water, mix them together, and you don't obtain a hurricane right away. So what you need to have to obtain this vortex-like wind structure is an energy gradient. So, for example, a temperature gradient or a pressure gradient across 100 or 1,000 kilometer scale. And only then, when you have these these conditions fulfilled, you obtain a process which is called the wind. So in the same fashion, we could approach metabolism. So we know all the building blocks of life, we know the elements that make up life, but you could take all the biomolecules that you know, you could put them in a container, stir with a spoon, and life will not happen. Because for life-like processes to emerge, you need to apply some sort of a gradient. And for the emergence of life, scientists have considered many types of gradients. For example, energy supplied by photochemistry or by electrochemical gradients. There are very many different approaches. But all in all, what it boils down to is that the release of this energy causes the structure, the self-organized structure to appear, and we call this structure metabolism, or before life proton metabolism. So we are chemists and we perform chemical experiments. So how do we choose what experiments to perform on what chemicals, and how do we even approach this problem from a chemistry point of view? So I would like to walk you through our thought process. In a living organism, we have established that the chemical processes that make up metabolism, they're promoted or they're catalyzed or facilitated by these complex structures that we call enzymes. So you have this rotating blue ribbon in the top right corner of the slide, and this is an enzyme. So obviously on the early Earth, these enzymes did not just float around, and they had not evolved yet. And another thing that we know about metabolic reactions is that many of these processes, they require energy. And biology uses an energy currency, which is called ATP most of the time. And the structure of ATP you have shown in the right bottom corner. And it is also quite unlikely that these complex molecules would have floated around in the prebiotic world. But what we know that was there on the early planet Earth is a lot of minerals and a lot of geochemical catalysts. So our approach to prebiotic chemistry is to draw a parallel between the way that these processes, different chemical processes, biochemical processes, are performed by life, and how they might have been performed by prebiotic chemistry before life had evolved, before enzymes, and before ATP was there. To help us understand which kind of biochemistry could have originated as prebiotic chemistry, it can be also helpful to make a different type of fundamental distinction between within life. So there are different types of metabolisms, and we can classify them into 4 major groups. So one division is whether a metabolism is autotrophic or heterotrophic. So whether the metabolism is contained within a single organism, or does it need to prey on other organisms, or whether it needs to acquire biomolecules from the environment to survive. Another distinction is whether the metabolism is oxidative or reductive. It's reductive if it acquires electrons from the environment. It is oxidative if it releases electrons to the environment. So many evolutionary biologists and chemists, like the guys shown in the slide, they support the view that the earliest metabolism was reductive and it was autotrophic. And why is that so? First of all, because most models indicate that the environment on the early Earth was mildly reducing, so it is likely that the metabolism was also relying on the electrons available from the environment. Another reason why they think that metabolism was in the earliest organisms, the metabolism was autotrophic, is because for heterotrophy, you would have to have very many organisms that inhabit a niche, so they can prey on one another. And that definitely was not the case when life was only emerging. So it has been proposed that reductive autotrophs, the metabolism from reductive autotrophic organisms, contains the most information about how the earliest biochemistry looked like. What I'm showing you here is a very simplified organizational scheme of such a metabolism. So it contains the most important classes of biochemical processes, of biomolecules, and what they all form is a giant biochemical network. So what happens here is that if you go from the left side of the slide, you see this little cloud with CO2. So what this metabolism does is it takes CO2, it converts it to a 2-carbon molecule called acetate, which then enters a cycle. So you have this circular shape. From there, the cycle, you can venture up toward either amino acids, which become converted into peptides and polypeptides, or you can go at the bottom of the scheme, you've got gluconeogenesis. So this is the chemistry that deals with sugars. So it builds up or breaks down sugars. And eventually, going to the right-hand side of the slide, we combine amino acids and sugars, and what we form is ribonucleotides, which then, if you go to the upper right corner, then we are starting to progress towards the genetic molecules and eventually entire genetic systems. I'm going to tell you something about most of these classes of reactions, most of these subsets. Some of this research is published, some of this is not published, and unfortunately I'll have to be a bit vague about the non-published stuff, but nonetheless, I think we'll touch on all of these reaction types or all of these network subsets. But before we do that, it's time for a definition. What is a reaction network? So a reaction network comprises two types of structures. So you have a picture of a network at the bottom left part, in the bottom left part of the slide. So a reaction network is a structure which where the nodes or the dots are different chemicals or chemical structures, and the connecting lines or vertices are the transformations between these two structures. So this is basically a chemical reaction network. From the point of view of prebiotic chemistry, it is important to make a distinction between discontinuous networks and the continuous approach to networks. A continuous approach is where each of these transformations is in one way or another kinetically coupled to the previous ones. And this continuous approach is an approach which allows you to change conditions or to make physical separations between different steps. So this continuous approach, as far as prebiotic chemistry goes, it has been criticized by several scientists because it allows for these step changes of conditions from one step to another. You can chemically purify after every step. You can change conditions, change pH. You can use a very well-timed additions of certain reagents or certain substrates. And if you don't follow these rules of those very well-timed additions, what you obtain is tar. And tar, we have this glove which is holding some like very, very gooey, horrible-looking substance that looks nothing like life. So now that we got the definition straight, we can go to actual chemistry, and I will be showing you chemical structures and actual chemical reactions. But if you are not a chemist, stay with me nonetheless, because you don't have to understand the chemistry on a molecular level to be able to understand how a process works, and I'll try to be very clear about that as well. So stay with me while I explain the general principle behind the entire processes. So here we are starting from CO2, like we've had on this previous slide. So in nature, there are 6 or 7, depends how you count, CO2 fixation pathways. That means pathways that convert CO2 to higher carbon molecules. And we know that they are used by microorganisms to build up biomolecules. So from the evolutionary standpoint, there are 2 carbon fixation pathways that are the most relevant. On the left-hand side of the slide, you have the Wood-Jungdahl pathway, or the acetyl-CoA pathway. So how it works is that it starts from CO2, it takes 2 molecules of CO2, and it converts them to a 2-carbon molecule of acetate. It is the only thermodynamically downhill pathway, and it is used by both bacteria and archaea, and it has also been postulated to have been present in Leuca. Leuca is the last universal common ancestor to all known life. And it is also the only linear pathway of carbon fixation that we know of. The other one is a bit more complicated. It is called the reductive Krebs cycle, or for short, the RTCA cycle, the reductive tricarboxylic acid cycle. I will use the term RTCA throughout this talk because it's the simplest. So what happens here, even if you are not a chemist, start from the 2-carbon molecule of acetate. So like in the center, the left side of the cycle. If we start from a 2-carbon molecule of acetate, we start to incorporate more CO2s, and we build up the carbon skeleton until we get to citrate, which contains 6 carbons. What happens then is citrate becomes broken into 2 molecules, so we have this loop. And this loop allows to produce another molecule of acetate and a 4-carbon molecule of oxaloacetate. The reductive Krebs cycle, the RTCA cycle, is very special because of this loop, but also because several molecules that are on this cycle allow for the production of different classes of other biomolecules. They are universal metabolic precursors. So acetate can give rise to, for example, fatty acids. Oxaloacetate can give rise to amino acids, and also it is used as a substrate eventually for the synthesis of ribonucleotides and another molecule. Succinate is a building block for cofactors, for porphyrins. And alpha-ketoglutarate allows us to synthesize amino acids as well. And pyruvate, of course, on the left-hand side, is a building block for sugars. So these 5 molecules are called the universal metabolic precursors to different other classes of biomolecules. Now, like you saw that on this previous slide, there is this loop. And we call this an autocatalytic loop, and the entire structure of the reductive Krebs cycle, the RTCA cycle, is autocatalytic. And what does that mean? From the system science perspective, we can say that the RTCA cycle is autocatalytic because with every turn of the cycle, the number of molecules doubles. So you get twice as many, 4 times as many, 8 times as many, 16 times as many, and so on and so on. How it works is that on the left-hand side, you have this circular shape, which also has an autocatalytic loop. And after every turn, of course, the molecules double. So this is called network autocatalysis. It works a bit differently to what we normally understand as catalysis in chemistry, because in chemistry we think about individual molecules that are catalysts. And here we don't forget about individual molecules, focus on the shape. So if you have one shape, it does one turn, at the end you get 2 of those shapes. So these shapes, we call them topologies. So the topology of the cycle allows it to self-amplify. The topology is the catalyst. So normally, for this structure on the left-hand side, if you want it to be sustainable, then every reaction requires at least a 50% yield. If this condition is fulfilled, then the cycle can self-amplify. But it is hard to imagine that on the prebiotic Earth you would have such efficient processes, because there are many side products, there are many, the geological environment is quite messy also. You have different detrimental, deleterious processes. And this would probably not be very practical to be used in a prebiotic world. So how to overpass that? There is an idea which involves a constant supply of one reagent. So on the right-hand side, you have this in the yellow box. One process supplies a reagent from the outside, and this leads to a structure which is called a stabilized network autocatalysis. So in this case, if you have one sort of feeder pathway that supplies one of the reagents to the cycle, in this case, it is enough for the individual reactions to have a yield just above 0%, and this is enough for this entire process to be self-sustainable. So following this idea, it has been hypothesized that the two carbon fixation pathways I just showed you on the previous slide, that they were once fused and they worked together as the stabilized network autocatalysis in the prebiotic world. So this is what it would have looked like. And we are chemists again, so we wondered, is it okay? Well, we know all these chemical structures. To chemists, these structures, they are not difficult, they're quite simple. So we wondered, is it possible that such a network would have worked without enzymes, just with mineral catalysts, or maybe without catalysts? We looked at it and thought, well, okay, on one hand, you might think that this is a bit unrealistic because CO2 by itself, it is not very reactive. But on the other hand, we looked at the enzymes themselves, and we noticed that many of these enzymes contain transition metals as cofactors, or transition metals in the active sites. And at the same time, we know that transition metals are used all the time as catalysts by synthetic organic chemists. So we thought, well, maybe we don't need enzymes. Maybe it's enough to have metals to do the same job. So we organized some pressure reactors as equipment. So you have this in the picture. And we decided to run some reactions with CO2 and different metal catalysts, metal powders. So if you're not chemists, maybe even if you are chemists and you haven't seen these pieces of equipment before, they're quite fun to work with. They are safe. They don't really need to, they don't really lead to evacuation of the lab, at least not normally, you would expect. And how they work is just like a regular pressure cooker in your kitchen. If you want to cook some, I don't know, beans or lentils, the same fashion you work with these pressure vessels. You put your reactions inside, then you use those nozzles to pump gas, you have a manometer to monitor for the pressure, and it is really straightforward. So what we did is some high throughput screening with different metals to see if any of those metals could promote the fixation of CO2 into higher carbon molecules. And we were especially curious about iron because iron was is very abundant geochemically. So that was one of my first slides, right? So we went on to do experiments, and after several hundreds or even thousands of experiments that we ran, we eventually noticed that simply heating some metals in salty water under the atmosphere of CO2, and the metals that we tested were molybdenum, iron, nickel, cobalt, manganese, or tungsten. So only heating these metals in salty water gave, we detected products, and the products were acetate, pyruvate, formate, and methanol. And this was quite a curious discovery because initially you could think that, okay, you're going to get a mess if you get different carbon fixation products. They will start to react with each other, and eventually you will end up with goo. And that was not the case. And even more interestingly, in the case of iron, of cobalt, and of nickel, we obtained a 3-carbon product, pyruvate. So all these products that we detected, they were literally following the topology of the Wood-Youngdahl pathway, so the first carbon fixation linear pathway that I showed you. We probed this chemistry a bit further, and we tested different temperatures, we tested different pressures, and just to cut this long story into a short one, the important thing is that this chemistry is very robust. It can work even at 30 degrees Celsius and under 1 atmosphere of CO2. And in this case, you only obtain 2 products, pyruvate and only obtain 2 products, acetate and pyruvate. Another thing we looked at is the product profile that we followed over time, and we didn't really know what that means in terms of the mechanism, but overall, what we found is that this reaction is, first of all, robust. You get products at a very large range of temperatures. And also, at different times, you get mostly acetate or mostly pyruvate. So this was very interesting to us. And also in some cases we noticed ethanol, so that was another extra product which we measured. As amazing as these results were, we also obtained some criticism. And these criticisms came from biologists and from geologists. Biologists pointed out that, okay, well, you obtained the Wood-Jungdahl pathway products, but the organisms that actually use this pathway to fix CO2, they never use iron as the electron source. They use hydrogen gas. And hydrogen gas is involved in a complicated process called electron bifurcation, which uses flavin cofactors, and it's very complex. At the same time, these organisms live in structures that we call hydrothermal vents. These are structures that you can find at the bottom of the ocean. They're very hot, they're hydrothermal chimneys, you have a lot of hot water, and you also have hydrogen gas, which comes from a geological process which is called serpentinization. So I'm not going to go into detail, but basically what happens, serpentinization is a process where you have hot water that comes in contact with a certain type of rocks, and as a result you have hydrogen gas which is produced. At the same time, geologists pointed out that, okay, you use metallic iron, but metallic iron was quite unlikely to have been lying around on the surface of the Earth, even of the early Earth, because right, it is abundant in the core of the planet, and it's also abundant into different types of space rocks. But on the surface of the Earth, and in the mantle and in the crust, you have more oxidized forms of the metal, and not really so much of metallic iron. So the evolutionary biologist who pointed this out to us was Bill Martin from Düsseldorf in Germany. And he has been pushing this hypothesis for about 20 years that the Wood-Jungdahl pathway would have been possible in hydrothermal vents. So we teamed up with Bill Martin and also with a heterogeneous chemistry specialist, Harun Tuisus from Mülheim, and also with microbiologists from Japan. We all teamed up and we did several, very many tests of different minerals. What we found was that if you use CO2 gas and hydrogen gas and 3 different hydrothermal minerals as catalysts, and these minerals are awaruite, greigite, and magnetite, which is basically rust. So these minerals allow for carbon fixation using hydrogen as an electron source, and you obtain the same type of products as you see in the Wood-Jungdahl pathway. So this finding brought us even closer to what actually biology does. We managed to mimic biology even better without any enzymes, and we proved that these processes can occur geochemically. If you understood absolutely nothing, or if I've been going through it too fast, or if you would like to read more about it, we have 2 blogs that have been published online. These are behind the paper blogs on the Nature, Ecology and Evolution website, the community contributions. So feel free to look them up, have a read, and these explain this research in a more simple language, perhaps. So I encourage you to look them up. Now, so can the primordial metabolism work without enzymes? The answer is yes for the left-hand side of the slide. So this we have established. Let's go on then. Now we will have a look at the reductive Krebs cycle, which is also called the RTCA cycle. So it runs in the counterclockwise direction. It has 11 reactions. 5 of these reactions require ATP, 6 do not. Yeah, we have this self-amplifying, this autocatalytic loop. Reactions are simple. So we also wanted to check whether maybe some or all of these reactions can be promoted by metals non-enzymatically. We were, of course, not the first ones to try to do that. There was a group of Scott Martin and co-workers who used the photocatalytic properties of manganese sulfides and zinc sulfides to try and catalyze some of the reductive Krebs cycle, RTCC cycle reductions. But the result of these investigations was such that only 2 reduction reactions that they tested gave yields above 3%, and they did not obtain continuous sequences that would allow them to perform the entire pathway in one go, for example. So in our work on the RTCC cycle, we started with the irreversible reductions, so you can see them as the magenta sort of red blobs. These are irreversible reductions. So we use these irreversible reductions to try and push the reversible hydration reactions, which are shown in green. So first of all, focus on the bottom part of the cycle. And after screening thousands of reactions manually, by hand, pipetting, and really working very, very hard, we found that the sequence of the bottom parts of the cycle, so starting from oxaloacetate, which is reduced to malate, which is then dehydrated to fumarate, which is then reduced to succinate, this sequence can be performed under acidic conditions with iron, metallic iron as a reducing agent, and with zinc salts. So this sequence, you plug in the starting material oxaloacetate, you add all the catalysts, all the promoters, you wait, and at the end you get succinate. The same worked for the upper part of the cycle. So we started with oxalosuccinate, which then becomes reduced to isocitrate, which then becomes dehydrated to aconitate, which then becomes rehydrated to citrate. So this upper arch of the cycle also can be promoted by iron and transition metal salts, so in this case also zinc salts and chromium salts, under acidic conditions as a one continuous sequence. So for what it's worth, it was probably, to the best of my knowledge, the first example of entire sequences of biochemical pathway that can be promoted non-enzymatically in one go, in one pot. Again, if this has been too confusing or too fast or too incomprehensible, I encourage you to look up the blog post where this is explained in a very, very high detail with pictures, using a language that is simple, and I hope it is understandable also for non-chemists. So again, the Nature, Ecology and Evolution blogs, please have a look. Now this is a short break, and I want to show you specifically what it means to do high throughput screening, what it looks like if you work on this type of chemistry in a chemistry lab. So we use lots of these small, tiny vials where we test hundreds, thousands of reactions under different types of conditions. So we do a deconvolution of different of these sets of conditions, and then we analyze. So two major ways to analyze these reactions in particular is the GCMS, so gas chromatography coupled to mass spectrometry, and also NMR, which is a standard chemical method for analyzing chemical reactions. So to recap quickly on where we are. So altogether, after screening nearly, I would say, 10,000 reactions, we found that the combination of metallic iron and transition metal salts allows us to promote all the ATP independent steps of both ancient carbon fixation pathways. So the acetyl-CoA pathway, the linear one on the left-hand side, and also two sequences of reactions from the Krebs cycle. So all those sequences that I told you about, they do not require ATP in nature. So we thought, okay, what is special about ATP? Why could we not promote these reactions? Why could we not find any sets of conditions that would make them go? What is the problem? How did prebiotic chemistry perform these reactions when there was no ATP? And we noticed, okay, well, look at them. The reactions that need ATP in nature, these are also the reactions that are carbon-carbon bond formations. And they are absolutely crucial, because if we want to perform anabolic processes that build up the carbon skeleton, we need to find a way for this to happen? How did prebiotic chemistry build carbon-carbon bonds if there was no ATP? And an important hint came from a paper in Cell by Daniel Gray. And in this study, this is a computational biology study. So what they did is they took the entire database of known biochemical reactions. So this database is called KEGG, K-E-G-G. And there are many databases like that. They took this one and they removed computationally all the reactions that depend on phosphate, because ATP contains phosphate. And what they obtained was quite curious, because it was a network of reactions. They were quite well connected. And the network looks like that, what you see in the slide. So how this works, you could compare it to a metro map. So if you have a map of a metro and you have different, those dots or those nodes are stations. So if a station is a very popular one, very central one, very busy one, it's usually a bigger node. And the lines are just trains that go to different directions and contain and connect the central stations to the smaller stations on the outskirts of a city. So the two stations, or the two nodes that were the most well connected in this metabolism that didn't depend on ATP, were two compounds. And these are these compounds, pyruvate and glyoxylate. Nowadays we can find them, for example, in apples. They're very abundant. So as chemists, we looked at these compounds and we saw, okay, well, these are carbonyl compounds. They can react with one another. And if they're so central, then perhaps the biochemistry, the early emerging biochemistry that did not depend on ATP, maybe it depended very heavily on these two compounds if they are so central in this network, in the simulation. Later, another paper came out by Springsteen, Krishnamurthy, and co-workers. And what they did was they showed that actually, well, these compounds do react together. And if you add an oxidant, they give rise to two cycles that they can, which can occur in one pot. And these two cycles, they produce two compounds of the Krebs cycle, oxaloacetate and malate. So we thought, well, this is quite a curious discovery, because what will happen if we add metals to this mix? We are very fond of metals, you might have noticed so far. And about a year and a half ago, we proposed a possible solution to how the CC bonds, the carbon-carbon bonds, would have formed in biochemistry that did not use ATP. So in this study, what we did is we reacted these two compounds together, pyruvate and glyoxylate, but we added iron salts in warm water. And we waited and we monitored what happens over time. So you wait 24 hours and you get 16 compounds. Some of them contain 6 or more carbons. And basically all of them are biologically relevant. So this was this made us really happy to discover that. So this graph that you see in the slide shows a mass balance where you see that the bigger the complexity, so the further you go to the right-hand side, the more mass you lose. And actually what happens is most of what we've noticed is that the mass becomes lost to CO2 in the major part. So now let's have a look at the network that we actually found. So we use different chemical methods, including isotopic labeling. I'm not going to talk about that. You can read the paper if you want. But basically what happens is you start from glyoxylate and pyruvate, which you should see in the yellow boxes. I cannot point on this now, but you should see in the left side of the slide, in the yellow boxes, you see the starting materials. And as time passes and you monitor what happens in this reaction, a mixture using analytical methods like the GCMS, you notice that you obtain more and more of the compounds which I have shown in this slide in this circular shape of a cycle, and also like different compounds that go a bit out of the cycle. Another thing that happens is that when you add hydroxylamine to this mixture and iron, you obtain 4 amino acids directly from this network at the same time: glycine, alanine, aspartic acid, and glutamic acid. So this is also quite interesting from the prebiotic chemistry point of view. 4 amino acids. To give you a better idea of what I'm talking about, why this is so important for biochemistry, for prebiotic chemistry, let's have a look at the same network, but this time it is overlaid on actual biochemical cycles, the ones that life uses even today. So what we see is on the left-hand side in red, you've got the Krebs cycle, and on the right-hand side in blue, with the blue colors, you have the glyoxylate cycle, which is used by plants. So the fat arrows highlight the reactions that are present in our network, and what we see is that in the case of the red cycle, 9 out of 11 intermediates of the biological Krebs cycle are contained within our network, the non-enzymatic one, and 7 out of 11 reactions, and also 8 out of 11 intermediates of the glyoxylate cycle, and 5 out of 8 reactions. So we are really starting to see and to recreate biochemistry without enzymes that really closely follows the transformations and the intermediates of the actual biochemistry that we see today. So this is great. And again, as always, if what I have said makes absolutely no sense and you would like to go through it again and maybe understand a bit better, I encourage you to look up the blog post. And also in this case, the American Society for Microbiology made a video about this system. So you can also look it up on YouTube. If you put American Society for Microbiology on the glyoxylate pyruvate network, I think you should find it quite easily. Before we move on, I would like to make a very important point. Because remember, I showed you initially when the complexity was growing in the pyruvate glyoxylate network, I also told you that we lose a lot of mass, and we are converting, also breaking down many of these molecules back to CO2. So this is bad, right? Well, the truth is that it's actually not so bad, and it might actually be needed, because the way it works is like this beautiful fountain in the center of Strasbourg. So you've got molecules that are built up, so the water goes up in the fountain, and then so at the same time, for you to notice that the shape of the fountain, the shape of water actually has a structure for you to see it, it also needs to fall down under gravity, so the molecules need to be broken down. And only when you have these two opposite processes that are maintained in a steady state, only then do you see the shape of the fountain architect of the shape that was initially designed by the fountain architect. So this is very important. It was probably actually very necessary that the build-up, so the anabolic and breakdown catabolic processes, to be linked by from the very beginning of prebiotic chemistry. So in other words, what needed to happen is that the proto-metabolism that emerged from geochemicals, it must have emerged in a formation destruction regime. So this is what systems chemistry scientists call this type of environment. So what this means for prebiotic chemistry, for the emerging proto-metabolism, is that you would have needed to have a constant supply of energy and of fuel, and also a constant presence of electron donors and electron acceptors. Now we're going to zoom out back to the big picture. So the green blob shows you how much we have actually covered so far, and there's a lot left. And I just told you that we need also destruction, so breakdown processes. So where do they actually fit in the big picture? So this has actually been achieved by another scientist. So in a study by Marcus Rauser and co-workers, it is shown that the oxidative Krebs cycle, so the the one that breaks down biomolecules to obtain energy, it can be promoted by peroxydisulfate under very, very familiar conditions. So you've got iron and you've got water at 70 degrees. So these conditions look familiar because they are exactly the same conditions except for the peroxydisulfate. But iron catalysis and water at 70 degrees, this is exactly what we used for the pyruvate and glycosylate network. So this actually made us happy when we saw that. What is actually left to be done, left to be dealt with in this simplified scheme of biochemical networks is this. So now I would like to tell you what the Moran lab, the laboratory of chemical catalysis, is working on at the moment. And most of this is not published, so I'm going to be slightly vague. Expanding towards the other non-enzymatic prebiotic chemistry means that we are also tackling sugar chemistry, so gluconeogenesis, the pentose phosphate pathway, and also the synthesis of ribonucleotides. So sugars are really important to life, and nowadays we use them to store energy and to fuel ourselves throughout our daily activities. But they are also important because they're the building blocks of genetic molecules, especially the ribose that we're going to see later. In prebiotic chemistry, one popular approach to building up sugar molecules is the Formose reaction, which uses formaldehyde as a building block, and under calcium hydroxide catalysis, it produces a mixture of pentose, so 5-carbon, and hexose, 6-carbon sugars. It's quite hard to control, but there are several methods that use minerals which bias this pathway towards pentose sugars. Another popular approach is the Kiliani-Fischer homologation of hydrogen cyanide, which is based on nitriles and hydrogen cyanide chemistry. But the truth is that life uses neither of these two reactions. Life uses a pathway called gluconeogenesis to build up sugar molecules. And most of the intermediates in the gluconeogenesis pathway contain molecules that are phosphorylated. So they contain phosphate groups on those intermediates. And this pathway starts from pyruvate. And after phosphorylation, you obtain a molecule which is called PEP, phosphoenol pyruvate. Then we've got a sequence of phosphorylations, isomerizations, reduction, and we end up with two important biomolecules, glyceraldehyde 3-phosphate, GAP, and DHAP, dihydroxyacetone phosphate. And you see them in the bottom right corner of the slide. These molecules undergo a crossed-Aldo reaction to give a sugar molecule. So we've got a pentose sugar, which then becomes converted into a hexose glucose. In our lab, there are two people who work on this project. A postdoc who has unfortunately just left, but she's found a really good job, so we're happy for her, and a PhD student, Joris. And I cannot tell you details yet, but given that in nature all these steps are promoted by enzymes and they also use ATP for phosphorylation, we have managed to find a geochemically abundant source of phosphate and several metal salts which promote most of these reactions. And at the moment we are working on unifying these conditions to take us from pyruvate and PEP following the individual steps of the gluconeogenesis pathway non-enzymatically to produce ribose, the pentose sugars, which we're later going to use in the synthesis of ribonucleotides. So here I must also mention the work of Markus Ralser again. So back in 2014, which is well before we have started to work on non-enzymatic analogs of biochemical pathways, these guys in the Ralser lab found quite serendipitously that a mixture of iron salts in the 70 degrees Celsius warm water can break down sugars following the pathway of glycolysis and also the pentose phosphate pathway. So they found all these intermediates that you can find on the slide. The black arrows correspond to glycolysis, while the blue arrows correspond to the reductive to the pentose phosphate pathway. And the important thing is that I hope you see this in the center of this scheme. You should see a ribose. So the ribose is the building block that we can use for the synthesis of ribonucleotides. So finally, ribonucleotides. The general skeleton of ribonucleotides is based on, like I told you, the ribose 5-phosphate sugar units, and we also have 4 nucleobases: pyrimidines, uracil, and cytosine, and also purines, adenine and guanine. So these nucleobases also have to be made in some way through prebiotic chemistry if we are to say that ribonucleotides could have been produced via non-enzymatic processes on the early Earth. So we've got these nucleobases and the sugar units. So let's go. How do we do that? To some prebiotic chemists, and there are quite many of them, the prebiotic synthesis of the building blocks of RNA is the only thing that matters for the emergence of life. So, of course, I'm coming here to talk about the metabolic perspective, so I don't entirely share this view, but on the other hand, at the same time, there is no doubt that RNA played a very important role, a key role in the origin of life, and it needs to be produced by prebiotic chemistry at some point, regardless of the scenario which came first, the other parts of metabolism or this part of biochemistry. So a lot of effort has been expended by prebiotic chemists in this direction, by brilliant chemists, and they produce brilliant pieces of research published in top journals. And I really, I'm not an organic chemist by training, so I'm in awe when I look at this, that they even came up with this. So we've got a synthesis by Sutherland on the left-hand side, and on the right-hand side we've got the synthesis by Carrel. So without going into very specific details, what I can tell you is that most of the time these approaches follow the discontinuous network approach, the one on the left a bit more so than the one on the right. And you've got, at some points, you have timed additions of reagents. On some instances, you have step changes of conditions. And in both cases, these syntheses use molecules that are foreign to biochemistry. Biochemistry does not use cyanoacetylene, for example. So as much as these are very impressive pieces of organic chemistry, we in our lab follow a bit of a different approach, and I think you can already imagine what it can be. So in biology, the synthetic strategy is quite different from the previous slide, and it's also a bit different for pyrimidine nucleobases compared to the purines. But both purines and pyrimidines, they are built from metabolites. So the carbon 1 unit, individual carbon units, are supplied by activated forms of formate of bicarbonate. Also the nitrogen and some parts of the carbon skeleton are supplied by amino acids. So they all come from other classes of metabolic pathways. Unfortunately, not that I know, but I don't think there are any reports of anybody who has ever tried to make ribonucleotides this way by using metabolites as opposed to the cyanide chemistry. So yeah, you should already be able to guess what our approach is. We also try to follow a pathway which is known from biochemistry. So I'm not going to go into very, very deep detail here, but in general, the biochemical pathway uses ribose 5-phosphate. It uses lots of enzymes. It uses 9 enzymes. It uses ATP for phosphorylation and glutamine as a source of ammonia, and also coenzyme Q as the oxidating to oxidize one of the key steps. So we in our lab started to work on trying to recreate this pathway non-enzymatically in a way that would conform to some sort of geochemical scenario. And without revealing details, because this is not published yet, I can already tell you that we also managed to identify a phosphate source which is available from geochemistry. We managed to identify some metal salts, minerals that promote most of the steps of this pathway. So we have had some disruptions thanks to COVID-19, but we're picking up, and if you stay tuned, you should soon see this research published. So this brings me to the end of my talk. Of course, this simplified biochemistry, largely based on carbon metabolism, after all, it's only a small part of the metabolic map. And this map only describes what we know about the life as we know it. But nonetheless, it is a great starting point for future explorations. And I would like to emphasize here and recapitulate on the point I made before on the hurricane slide, I believe. So in the living organisms, the metabolic pathways need to work together as a whole. What this means for us as prebiotic chemists is that any sort of constraints that we apply to non-enzymatic chemistry of one reaction or of one pathway, they also start to become relevant to others, because everything needs to remain in a state where they are interdependent or kinetically coupled. So we need to keep this in mind. We cannot treat subsets of this metabolic maps as something that happens in one container and then we magically jump to another one. So this is important to consider. Before I finish, I would like to quickly touch on the future perspectives for prebiotic chemistry. So at this point, we have talked about minerals that are used as catalysts to promote pathways which start to look like biological pathways in terms of the transformations or in terms of the intermediates. But if we look at the catalyst itself, how did we even go from something that is a mineral or a piece of rock? How did we jump, or how did we make this transition in biochemistry that was just developing to the biochemistry that we have now that uses complex enzymes? So I think a very important part of future research will be to try and look into biocatalysts, something in between, something that's more complex than a mineral, but much less complex than this insanely convoluted protein that you have rotating in the black part of the slide. So in my view, this is going to a very important aspect of biochemical of this prebiotic chemistry research. And apart from the exploration of the evolution of biocatalysis through prebiotic chemistry, other important aspects, I think, will touch on the evolution of genetics, but also its relationship with amino acids. So we still don't know why it is that amino acids are coded by nucleobases, how this process came to be so closely intertwined. And also there is a question of homochirality. Why does life use only D sugars but L amino acids? How did this come about? And finally, another important aspect of the prebiotic chemistry that led to life's emergence is looking at the behavior and the emergent properties of very complex systems that contain dynamic processes and messy mixtures, and trying to see what happens if you look at these very complex processes as they happen. To summarize, the main conclusion is that you should remember from this lecture is that non-enzymatic analogues of core biochemical pathways may have preceded the biological enzymatic ones. And secondly, these non-enzymatic protometabolic pathways can be promoted by transition metals. And these transition metals, such as iron, were likely very easily available on the young planet. Of course, there is still a number of fundamental questions that we still haven't answered, and we are very far away from discovering how life emerged on the early planet. But what is important to know, I think, is contained in this quote from Nick Lane, that life itself is the best guide to uncovering its own origins. And I think this is something that we have to keep in mind while we are trying to solve this problem. So before I really finish, I would like to advertise an initiative which is an Origin of Life network for early career scientists. And by early career, I mean anybody who is interested as a student, as a bachelor student, as a master student, as a PhD student, or a postdoc. This is a very friendly network which was established by several friends of mine who are also Origin of Life scientists. So I encourage you to go to oln.org and have a look. And we organize meetings and conferences and workshops. And I think if you're into the Origin of Life research, this is something that you might enjoy. Finally, acknowledgements. First of all, I thank you for your attention, for staying with me for over an hour. But also, I would like to thank all those people who are behind this research, because I think it's very clear that it's not me single-handedly who has carried out thousands of reactions and discussed the problems, came up with ideas. First of all, I would like to thank the director of our lab, the Lab of Chemical Catalysis, Professor Joseph Moran, who has been a great mentor for me over 5 years already. And all the co-workers who contributed, especially Dr. Srijit Varma, who is an author of most of the papers I presented to you today, and he did fantastic work, and I wish all the best to him. And also all the best to you. Thanks for your attention, and I will be happy to answer questions.